This video is about endocrinology, the introduction and some other aspects, organization of the endocrine system. So, with the help of some video lectures, we are going to learn about the endocrine system in detail, organization of the endocrine system, hormone receptor interactions, anterior pituitary gland and hypothalamus, posterior pituitary gland, thyroid gland, calcium homeostasis, adrenal cortex and adrenal medulla, and endocrine pancreas and glucose homeostasis. All these aspects we won't be learning. Some topics we will be covering with the help of other videos. So in this video we are going to learn about the organization of the endocrine system as a whole. So we will be defining the term hormone, defining the terms paracrine and autocrine, and some examples. Describe the location of the major endocrine organs, illustrate the levels of cortisol within the endocrine system, then describe the concept of negative feedback with examples. So let's start. Before learning about this topic, let's learn some important terms or jargons, technical terms related to this topic. Endocrine gland. What is an endocrine gland? These are glands that secrete their hormones directly into the blood rather than through a duct. And prostaglandins are a family of lipid compounds found in various tissues and are involved in a wide range of body responses, including endocrine responses, inflammation, muscle contraction, and homeostasis. The human body has two major control mechanisms, the nerve system and the endocrine system. While the nerve system can control and communicate rapidly with other systems, the endocrine system controls and communicates with the help of chemical messengers and is a much slower system. However, the effects of the endocrine system may be a more prolonged and sustained. Definition of a hormone. The term hormone is derived from a Greek word meaning to excite or to arouse. The term was first used by Ernest Henry Sterling in 1905 in his first lecture delivered to the Royal Society. Sterling, along with Bayliss, had discovered one of the first hormones called secretin in 1902. The endocrine hormone is a chemical substance that is produced by endocrine glands or a group of endocrine cells in response to certain stimuli and carried by blood to target tissues where they exert their physiological actions. The endocrine hormones are carried by the circulatory system to cells throughout the body, including the nerve system in some cases, where they bind with receptors and initiate many cell reactions. Some endocrine hormones affect many different types of cells of the body. For example, growth hormones from the anterior pituitary gland cause growth in most parts of the body, and thyroxine from the thyroid gland increase the rate of many chemical reactions in almost all the body's cells. Coordination of body functions by chemical messengers. The multiple activities of the cells, tissues, and organs of the body are coordinated by the interplay of several types of chemical messenger systems. Like neurotransmitters are released by axon terminals of the neurons into the systemic junctions and act locally to control nerve cell functions. Endocrine hormones are released by glands or specialized cells into the circulating blood and influence the function of target cells at another location in the body. So neuroendocrine hormones are secreted by neurons into the circulating blood and influence the function of target cells at another location in the body. Paracrines are secreted by cells into the extracellular fluid and affect neighboring target cells of a different type. Autocrines are secreted by cells into the extracellular fluid and affect the function of the same cells that produce them. Cytokines are peptides secreted by cells into the extracellular fluid and can function as autocrines, paracrines, or endocrine hormones. Examples of cytokines include the interleukins and lymphokines that are secreted by helper cells and act on other cells of the immune system. Cytokine hormones, for example leptin, produced by adipocytes, are sometimes called adipokines. In the next few chapters, which we will be learning with the help of various videos, we discuss mainly the endocrine and neuroendocrine hormone systems, keeping in mind that many of the body's chemical messenger systems interact with one another to maintain homeostasis. For example, the adrenal medulla and the pituitary gland secrete their hormones primarily in response to natural stimuli. The multiple hormone systems play a key role in regulating almost all body functions, including metabolism, growth and development, water and electrolyte balance, reproduction, and behavior. For instance, without growth hormone, a person would be a dwarf. Without thyroxine and triiodothyronine from the thyroid gland, almost all the chemical reactions of the body would become sluggish and the person would become sluggish as well. Without insulin from the pancreas, the body cells could use little of the food carbohydrates for energy. And without sex hormones, sexual development and sexual functions would be absent. Paracrine secretions. Paracrine secretions are those chemicals, when released, exerts its effects on cells and tissues located in the neighborhood of its site of secretion. This, by convention, includes neurotransmitters. Paracrine hormones are dispersed by simple diffusion in the interstitial fluid, and this restricts their action to short distances. So local enzymes in the vicinity usually rapidly inactivate the paracrine secretions, that preventing adequate entry into the bloodstream. Examples of paracrine secretions include the following. Histamine released from cells in the stomach mucosa has a direct influence on the parietal cells of the oxyntic glands, causing an increase in acid secretion. The neurotransmitters such as acetylcholine act on the parietal cells in the stomach mucosa, causing an increase in acid secretion. Then prostaglandins, PGE2, is responsible for cervical dilation in the time of childbirth or parturition time. Next, autocrine secretion. Autocrine secretions are chemicals which are secreted by specific cells into the extracellular fluid surrounding the cell and which act upon the very cell that secrete it. Examples of autocrine secretions are as follows. Norepinephrine, that is released by neurons in the adrenal medulla, further inhibits non-epinephrine released by those cells. So insulin released from beta cells in the pancreas has an inhibitory effect on insulin secreted by these beta cells, which is independent of glucose levels in the blood. So location of major endocrine organs or anatomical loci of the major endocrine glands and endocrine tissues of the body, except for the placenta, which is an additional source of sex hormones. Feedback control of hormone secretion, levels of control within the endocrine system. So we just mentioned about some points, some other aspects we are going to learn. Levels of control within the endocrine system. Although the plasma concentrations of many hormones fluctuate in response to various stimuli that occur throughout the day, all hormones studied thus far appear to be closely controlled. There are several control mechanisms that exist to regulate the levels of hormones very precisely. An example to review is the hypophysiotropic hormones, with a 3-level sequence to understand the different levels of hormonal control. So endocrine glands, hormones and their functions and structure. Major glands include hypothalamus, anterior pituitary, posterior pituitary, thyroid, adrenal cortex, adrenal medulla, pancreas, parathyroid, testis, and ovaries. There are 3 levels of feedback control as indicated. Long loop feedback control. In this case, the hormone secreted by the peripheral endocrine gland provide a negative feedback to the hypothalamus or the anterior pituitary gland. For example, excess levels of thyroxine will inhibit both the anterior pituitary gland from secreting thyroid stimulating hormone and the hypothalamus from secreting thyrotropin releasing hormone. Short loop feedback control. In this mechanism, the anterior pituitary hormone in plasma provides a negative feedback to the hypothalamus, decreasing the secretion of releasing hormones. Then ultra short loop feedback control. In this mechanism, the releasing hormone themselves have a negative feedback influence on the hypothalamus, thus decreasing the secretion of the hypothalamic hormones. Negative feedback prevents overactivity of hormone systems. In most instances, hormonal control is exerted through negative feedback mechanisms that ensure a proper level of hormone activity at the target tissue. After a stimulus causes release of the hormone, conditions or products resulting from the action of hormone tend to suppress its further release. In other words, the hormone or one of its products has a negative feedback effect to prevent over secretion of the hormone or overactivity at the target tissue. The controlled variable is sometimes not the secretory rate of the hormone but the degree of activity of the target tissue. Therefore, only when the target tissue activity rises to an appropriate level will feedback systems signals to endocrine glands become powerful enough to slow further secretion of the hormone. So, feedback regulation of hormones can occur at all levels, including gene transcription and translation steps involved in the synthesis of hormones and steps involved in processing hormones or releasing stored hormones. So CNS stimulus, hypothalamus, releasing hormone in plasma, anterior pituitary gland, anterior pituitary hormone in plasma, peripheral endocrine gland, peripheral endocrine hormone in plasma, target tissue for level 3 hormones. Hypothalamus, thyroxine releasing hormone, anterior pituitary gland, thyroid stimulating hormone, thyroid gland, triiodothyroidine, thyrosin in plasma, target tissue. So examples of negative feedback mechanisms. Negative feedback by plasma levels of thyroid hormones on the hypothalamus and the anterior pituitary gland. Negative feedback by plasma cortisol level on the hypothalamus and the anterior pituitary gland. Then surges of hormones can occur with positive feedback. In a few instances, positive feedback occurs when the biological action of the hormones causes additional secretion of the hormone. An example of positive feedback is the surge of luteinizing hormone that occurs as a result of stimulatory effect of estrogen on the anterior pituitary before ovulation. The secreted LH or luteinizing hormone then acts on the ovaries to stimulate additional secretion of estrogen, which in turn causes more secretion of LH. Eventually, LH reaches an appropriate concentration and typical negative feedback control of hormone secretion is then exerted. Then clinical variations occur in hormone release. Superimposed on the negative and positive feedback control of hormone secretion are periodic variations in hormone release that are influenced by seasonal changes, various stages of development and aging, and diurnal cycle or daily cycle and sleep. For example, the secretion of growth hormone is markedly increased during the early period of sleep, but is reduced during the later stage of sleep. In many cases, this cyclical variations in hormone secretion are due to changes in activity of neural pathway involved in controlling hormone release and clinical conditions associated with altered hormone release. Various clinical conditions can manifest if the secretion of hormones is altered either due to hyposecretion or hypersecretion. Hormone receptor interactions. Next topic, hormone receptor interactions. Here we are going to classify the various types of hormones, describe the hormone secretion, transport and clearance from the blood, classify the various types of receptors, describe the hormone receptor interactions in terms of upregulation and downregulation, define second messenger and list the various second messenger systems, differentiate between genomic and non-genomic effects of hormones. Some major terms, catecholamines. They are a group of amines that include epinephrine, norepinephrine, and dopamine and have important psychological effects as neurotransmitters and hormones. Downregulation, the decrease in the number of receptors in response to the high concentration of circulating hormones is referred to as downregulation. Ligand, it is derived from the Latin word ligandus, meaning to bind. A ligand could be an ion, a molecule, or a hormone that binds to another chemical entity or a receptor to form a larger complex. Then upregulation, an increase in the hormone receptor number as a consequence of sustained low exposure to a particular hormone called upregulation. Chemical structure and synthesis of hormones. Hormones can be classified based on their origin or by their chemical structures. There are three general classes of hormones exist: proteins and polypeptides. Proteins and polypeptides, including hormones secreted by the anterior and posterior pituitary gland and pancreas, insulin and glucagon, and parathyroid gland, parathyroid hormone, and many others. So, steroids secreted by the adrenal cortex, cortisol and aldosterone, and ovaries, estrogen and progesterone, and testosterone and placenta, estrogen and progesterone. So, derivatives of the amino acid tyrosine secreted by the thyroid, thyroxine, and triiodotyrosine, and the adrenal medulla, epinephrine and norepinephrine. So, polypeptide and protein hormones are stored in secretory vesicles until needed. Most of the hormones in the body are polypeptides and proteins. These hormones range in the size from small peptides, which as few as 3 amino acids like thyrotropin releasing hormone, to proteins with almost 20 amino acids, growth hormone and prolactin. In general, polypeptides with 100 or more amino acids are called proteins, and those with fewer than 100 amino acids are referred to as peptides. Protein and peptide hormones are synthesized in the same fashion in most other proteins. Steps in synthesis of protein and peptide hormones is as follows. First, synthesized as large biologically inactive proteins, that is pre-pro hormones. First, synthesized as large biologically inactive proteins, pre-pro hormones, cleaved to form smaller prohormones in the endoplasmic reticulum, then transferred to the Golgi apparatus for packaging into secretory vesicles. Enzymes in the vesicles cleave the prehormones to produce smaller biologically active hormones and inactive fragments. The vesicles are stored within the cytoplasm until their secretion is needed. Secretion of the hormones as well as the inactive fragments into the interstitial fluid or directly into the fluid's bloodstream occurs by exocytosis. In many cases, the stimulus for exocytosis is increased cytosolic calcium concentration caused by depolarization of the plasma membrane. In other instances, stimulation of an endocrine cell surface receptor causes increased cyclic adenosine monophosphate CAMP and subsequently activation of protein kinase that initiates secretion of the hormone. The peptide hormones are water soluble, allowing them to enter the circulatory system easily, where they are carried to their target tissue. So, steroid hormones are usually synthesized from cholesterol and are not stored. The chemical structure of steroid hormones is similar to that of cholesterol, and in most instances, hormones are synthesized from cholesterol. They are lipid soluble and are structurally related to cholesterol. Although there are usually very little hormone storage in steroid producing endocrine cells, large stores of cholesterol esters in cytoplasm vacuoles can be rapidly mobilized for steroid synthesis after a stimulus. Much of the cholesterol in steroid producing cells comes from the plasma, but there is also de novo synthesis of cholesterol in steroid producing cells because they are highly lipid soluble. They can simply diffuse across the cell membrane when synthesized and enter the interstitial fluid and then the blood. Then amine hormones are derived from tyrosine. The two groups of hormones derived from tyrosine are as follows: the thyroid hormones and the adrenal medullary hormones, catecholamines. The thyroid hormones are synthesized and stored in the thyroid gland and incorporated into macromolecules of the protein thyroglobulin and is stored in large follicles until secretion. After entering the blood, most of the thyroid hormones combine with plasma proteins, especially thyroxine binding globulin, which slowly release the hormones to the target tissues. So, epinephrine and norepinephrine are formed in the adrenal medulla. So, catecholamines are taken up into performed vesicles and stored until secreted and released by exocytosis. Once the catecholamines enter the circulation, they can exist in the plasma in a free form or in conjugation with other substances. Hormone secretion, transport, and clearance from the blood. hormone secretion after the stimulus, and duration of action of different hormones. Some hormones such as norepinephrine and epinephrine are secreted within seconds after the gland is stimulated and may develop full action within another few seconds to minutes. The actions of other hormones such as thyroxine or growth hormone may require months for full effect. Thus, each of the different hormones has its own characteristic onset and duration of action, each tailored to perform its specific control functions. So, concentrations of hormones in the circulating blood and hormonal secretion rates. The concentration of hormones required to control most metabolic and endocrine functions are incredibly small. Their concentrations in the blood range from as little as 1 pg, which is 1 millionth of the 1 millionth of a gram, in each milliliter of blood up to at most a few micrograms, a few millionths of a gram per milliliter of blood. Similarly, the rates of secretion of the various hormones are extremely small, usually measured in micrograms or milligrams per day. Then transport of hormones in the blood. Water soluble hormones. Peptides and catecholamines are dissolved in the plasma. They are transported from their sites of synthesis to target tissues. They diffuse out of the capillaries into the interstitial fluid and ultimately to target cells. Steroid and thyroid hormones. These hormones, in contrast, circulate in the blood, mainly bound to plasma proteins. Usually, less than 10% of steroid or thyroid hormones in the plasma exist free in solution. More than 99% of thyroxine in the blood is bound to plasma proteins. However, protein-bound hormones cannot easily diffuse across the capillaries and gain access to their target cells and are therefore typically inactive until they dissociate from plasma proteins. The relatively large amounts of hormones bound to proteins serve as reservoirs, replenishing the concentration of free hormones when they are bound to target receptors or lost from circulation. Binding of hormones to plasma protein greatly slow their clearance from the plasma. So, clearance of hormones from the blood. Two factors can increase or decrease the concentration of hormone in the blood: the rate of hormone secretion into the blood, the rate of removal of the hormone from the blood, metabolic clearance rate. Then metabolic clearance rate. This is usually expressed in terms of a number of milliliters of plasma cleared of the hormone per minute. To calculate this clearance rate, one measure of the rate of disappearance of the hormone from the plasma and the plasma concentration of the hormone, nanograms per milliliter of plasma. Then the metabolic clearance rate is calculated by the following formula. Metabolic clearance rate is equal to rate of disappearance of hormone by rate of disappearance from the plasma, rate of disappearance of hormone from the plasma by concentration of hormone, rate of disappearance of hormone from the plasma by concentration of hormone. Hormones are cleared from the plasma in several ways, including metabolic destruction by the tissues, binding with the tissue, excretion of by the liver into the bile, and excretion by the kidneys into the urine. For certain hormones, the decreased metabolic clearance rate may cause an excessively high concentration of the hormone in the circulatory body fluid. For example, when the liver is diseased, the steroid hormone levels increase because these hormones are conjugated mainly in the liver and then cleared into the bile. Most of the peptide hormones and catecholamines are water soluble and circulate freely in the blood. They are usually degraded by enzymes in the blood and tissues and rapidly excreted by the kidneys and liver, thus remaining in the blood for only a short time. For example, the half-life of angiotensin 2 circulate in the blood is less than a minute. So, mechanisms of action of hormones, hormone receptors and their activation. The first step of a hormone's reaction is to bind to specific receptors at the target cell. Cells attack receptors for the hormones to not respond, and the hormone combines with its receptor. This action usually initiates a cascade of reactions in the cell, with each stage becoming more powerfully activated so that even small concentrations of the hormone can have a large effect. Hormone receptors are large proteins, and each cell that is to be stimulated usually has some 200 to 10,000 receptors. Also, each receptor is usually highly specific for a single hormone. Each determine the type of hormone that will act on a particular tissue. The target tissue that are affected by the hormone are those that contain its specific receptors. So, hormone receptors have been discussed later. So, hormone receptor interactions, the number of sensitivity of hormone receptors are regulated. The number of receptors in a target cell usually does not remain constant from day to day or even from minutes to minutes. Receptor proteins are often inactivated or destroyed during the course of their function, and at other times they are reserved, reactivated, or new ones are manufactured by the cell. For instance, increased hormone concentration and increased binding with its target cell receptors sometimes cause the number of active receptors to decrease. Then downregulation of receptors. The decrease in the number of receptors in response to a high concentration of circulating hormone is referred to as downregulation. So downregulation of the receptors can occur as a result of inactivation of some of the receptor molecules, inactivation of some of the intracellular protein signaling molecules. Temporary sequestration of the receptor to the inside of the cell away from the site of action of hormones that interact with the cell membrane receptors. Disruption of receptors by lysozymes after they are internalized or decreased production of the receptors. At each case, receptor downregulation decreases the target tissue's responsiveness to the hormone. And upregulation of receptors. An increase in the hormone receptor number as a consequence of sustained low exposure to a particular hormone is called upregulation. Some hormone cause upregulation of receptors and intracellular signaling proteins. That is, the signaling hormone induces greater than normal formation of receptor or intracellular signaling molecules by the target cell or greater availability of the receptor for interaction with the hormone. When upregulation occurs, the target tissue becomes progressively more sensitive to the stimulatory effects of the hormone. Then, intracellular signaling after hormone binding activation, almost without exception, a hormone affects its target tissue by first forming a hormone receptor complex. Formation of this complex alters the function of the receptor, and the activated receptor initiates the hormone effects. The various mechanisms of hormone receptor interactions are listed and explained as follows: ion channel linked receptors, G protein linked hormone receptors, enzyme linked hormone receptors, and intracellular hormone receptors. Ion channel linked receptors. Virtually all the neurotransmitter substances such as acetylcholine and norepinephrine combine with the receptors in the postsynaptic membrane. This combination almost always causes a change in the structure of the receptor, usually opening or closing a channel for one or more ions. Some of these ion channel linked receptors open or close channels for sodium ions, others for potassium ions, others for calcium ions, and so forth. The altered movement of these ions through the channels cause the subsequent effects on the postsynaptic cells. G protein coupled receptors, enzyme linked hormone receptors are some other aspects which we will be learning tomorrow. Thank you for listening.